Okay, sir. I will activate now.
Okay. Oh, sorry. The live is going. Okay.
Yo. Okay, sir. I will activate now. Yeah. Sorry for the delay. We are almost come to the end of the 3 days Indo-UK virtual conference. Now we have a keynote talk will be delivered by Professor Thomas Webster, Chair, Professor of College of Engineering, Northeastern University, Boston. Welcome you, sir. I would like to welcome Professor Geeta Manivasagam, Director, CBCMT, Vellore Institute of Technology. I welcome you, ma'am. Please take over the session, ma'am.
Thank you, Dr. Loganathan. Good morning to Thomas Webster and good evening to all the participants. Before we start the session, I would like to share the good news about the prize winners of our conferences who are presented, had an oral presentation and poster presentation. Dr. Loganathan? Yeah. So the prize winners are the Shivaji Kaste, Department of Stem Cell and Regenerative Medicine, and his title was Layer by Layer Decorated Herbal Cell Compatible Scaffolds for Bone Tissue Engineering, a Synergic Effect of Graphene Oxide and Scissors Chondrologists. Congratulations, Shivaji. And the next is Aditya Jain from Center for Biosystem Science and Engineering, Indian Institute of Science. Her topic was In Vitro Disease Model for Skeletal Muscle Degeneration. The third is Kamini MD, the doctor, Center for Biosystems and also from Indian Institute of Science. And the next winner is Unikrishnan BS, Laboratory of Biopharmaceuticals and Nanomedicine Division of Cancer Research, RCC Trivandrum, and his topic was folic acid conjugated polysaccharide iron oxide nanoparticles, a new platform for magnetic field mediated liquid biopsy. And the final winner is Yoti Prabhunath. He is a professor from Oral and Maxillofacial Pathology, Vivekananda Dental College for Women, Tamil Nadu, India. Neurophotofins and neurophotofin receptors and emerging therapeutic paradigm in amyloblastoma tumor. So congratulations to all winners. Now we move on to our special keynote talk. The best poster presentation winners are Sumaira Malik, Amity Institute of Biotechnology, Ranchi, Hagmadin Khan, Center for Nanotechnology Research, VIT Vellore, Navpreet Kaur, Discipline of Bioscience and Bioengineering, IIT Indore, and P. Baladhan Mugam, Center for Nanoscience and Technology, Innova University, Chennai. So congratulations to all winners. I don't think like anybody the researchers working in the field of nanomaterials and biomaterials need an introduction for Professor Thomas. He's basically a chair and professor from chemical engineer, my long-term collaborator and friend. He is a head for nanomedicine laboratory. His research interest includes design, synthesis, and which he evaluates for the different applications, whatever applications nanomaterials has. He also directly or indirectly directs some centers in area of biomaterials, which is in Colombia, and 2 centers in China. He has a big group. Always there are around 40 to 60 students in his group, starting from undergraduate to postdoc researchers. And this group has extensively published, and they have generated around 13 textbooks and 68 book chapters. And since no nanomaterial conferences and biomaterial conference go without him, he has presented 276 keynote and invited presentation, and he has around 600 literature review articles published, and along with conference proceedings, which is around 700. His credit is mainly, he focuses more on the translation research, and he has published 42 patents, provisional or full patents, and Out of these patents, 9 companies have emerged. So as I said earlier, he's a chief editor-in-chief of International Journal of Medicine, and also is there in 15 additional journals in the editorial board. So he has organized several conferences and symposia. I don't want to stand between the participants and Thomas because everybody is waiting to hear from him. Over to you, Thomas. I now invite you to present your lecture on nanomedicine advanced for COVID-19. Welcome, Tom.
Yeah, thank you very much, Geetha, and it's a wonderful opportunity to see everybody on Zoom here and talk about science and talk about the things that we can do in terms of this current pandemic. I don't know if I am sharing my screen. Good, so everything looks good, right?
Yeah.
So it's certainly wonderful. I can't wait for the time though that I can get on a plane and come to Vellore and VIT and see everybody. So hopefully that will be shorter rather than longer. But yeah, so thank you very much for this opportunity to talk about what we're doing in nanotechnology related to COVID-19. I am going to be presenting some work that has recently been licensed to a company, most notably this company Audix, listed here. They are licensing some of our nanomaterials for therapies in terms of COVID-19. So I want to give that disclosure. But really, I want to start talking about what this pandemic has done to us, to the globe. And I think one of the issues that I'm often reminded of as we go through COVID-19 is how it has impacted our healthcare system. And I think really it has brought to light some problems in our healthcare system, some problems that already existed that are just now amplified because of COVID-19. I think these first two bullets are important for what we do, for what COVID-19 has brought out in our global healthcare system. And this is true, I think, whether you talk about what's going on in India or what's happening in the United States. I think clearly our hospitals have been overwhelmed with patients who want to know if they have COVID-19, and certainly for those patients who have symptoms for COVID-19 to go and seek treatment. And I think our hospitals cannot handle, they have not been able to handle the large number of patients that are suffering from COVID-19. And I think what has also been emerging with COVID-19 is there are now a certain subset of people who are afraid to go into the hospital for fear of catching COVID-19. We see a lot in the United States, a high number of people who might have trouble with their heart rate, or they might have trouble with managing diabetes, or whatever their health problem is. They don't want to go to a hospital where you have infected patients for fear of catching COVID-19. So I think, at least in these two bullets, overwhelmed hospitals, barriers to entering a hospital or a doctor's office, we can use nanomedicine to reduce those problems. So, some 25 years ago, we started looking at nanomaterials, we started looking at nanoparticles to reduce the need for somebody to go into a hospital, to reduce that barrier that people have of seeking help. And, I think we often talk about this, but as an introduction to how we're using nanomaterials for COVID-19, we have looked a lot at our background, at how we have used nanostructured materials, nanoparticles to control cell responses. And we've learned a lot through the years. We've learned that nanotextures, as I think some of you have also learned, can increase bone growth. They can increase function of an amputee device, increase vascularization for stents or polymers that have nanostructured features. They can also be used to reduce infection. without using antibiotics or drugs. And also we have seen that they can increase orthopedic soft tissue growth, like increase ligament attachment or regrowth, increase cartilage regrowth. So highlighted on this slide are just various examples of how we've used nanostructured surfaces, nanomaterials in terms of controlling cell responses. And our group has focused on doing so without drugs, without antibiotics, or without anti-inflammatories. We've been really trying to develop materials that have properties that can increase tissue growth, control cell responses without using drugs. And of course, this has been wonderful. I've been so happy to be a part of these types of research areas. But today, we really have a different problem we want to talk about. And that obviously has to do with viruses. Can nanomaterials control cell responses, or I should say, can nanomaterials control virus responses in the same way that we have seen them control cell and bacteria responses? And we're seeing the answer is yes. So we get pretty excited about this. But I really would like to make the argument to, my fellow colleagues and scientists that we should have seen this coming. Virus infections are not something that's new. And in fact, you can see in the middle of this slide a couple of viruses that we are all too familiar with. MERS, the Middle Eastern respiratory syndrome, norovirus. Norovirus was a huge problem in the United States several years ago at some restaurants where the virus adsorbed to surfaces and they were able to infect other people. So COVID-19 is really another virus in a long list of viruses that have plagued humans, have plagued our healthcare system. And, I don't think I'm making too much of a risk by saying after we get past COVID-19, there will be another one. There's going to be another virus that we're going to have to deal with. I think another thing that I often talk about that is on the horizon that is associated with viruses are bacteria infections. And we are all well aware of what's happening, and I've highlighted on this slide, by 2050, a number of places have predicted the significant impact bacteria infections, antibiotic-resistant bacteria infections will play on our healthcare system. So we need to start preparing now. And when I look at what's happening with COVID-19, when I look at what's happened with these prior viruses, we have to change fundamentally how we are treating these problems. So I make the argument, despite that COVID-19 is new, it is a new virus that is infecting us. We should have seen it coming, and we should have been able to adjust our healthcare system appropriately to reduce our dependence on hospitals, to reduce our dependence on drugs that are used for treating bacteria infections. All of these things we have to start thinking about and changing. And you can see on this slide where our traditional approach to fighting bacteria, to fighting viruses has not changed in decades. And in fact, I'm reminded of this COVID-19 virus as soon as it was identified, as soon as we knew it was going to be a global pandemic, immediately everybody said, oh, well, there'll be a vaccine. We will be able to generate a vaccine that in a year and a half, COVID-19 won't be a problem. Well, we're in that period of waiting for a vaccine. We have to change the way our healthcare system looks towards these traditional solutions, because we have seen what happens right now when that occurs. Countries shutting down, travel shut down, people losing their jobs. In the United States right now, our unemployment is approaching 20%. The old way of fighting a virus, or the old way of fighting even bacteria, has to change. And of course, I think nanotechnology might be the answer, just like it was the answer for us to increase tissue growth by using nanostructured features, and just as it is being used to fight bacteria infections, can nanotechnology be used for viruses? And let me go through some reasons why I think that answer is yes. First and foremost, all viruses are nanostructured. Norovirus that I mentioned before, which created problems for our restaurants, is nanostructured. COVID-19 is nanostructured. It has, of course, a membrane protein. It has spikes. We often, we call these S proteins. These S proteins are what attaches to a cell, enables the virus to go into a cell, and then releases the RNA to divide. These are nanostructures. So we know that. And I, make the argument, in order to fight a nanostructure like a virus, you need another nanostructure like a nanoparticle or something else. The other thing we know about COVID-19 is the second bullet listed on this slide. It has multiple nanostructured functionalities which can be targeted. And here, it's very important for us to think not just targeting the S protein. The S protein is very important, as we all know, for coronaviruses and COVID-19 to attach to a cell membrane. And it's what we would automatically think to develop a strategy to block those S proteins from attaching to a cell. But guess what else happens for S proteins in a lot of coronaviruses? They mutate. So if your only strategy is to develop a molecule, develop a nanoparticle like we are doing to attach to the S protein of COVID-19, and it mutates, your strategy now won't work. So the other thing we can do a lot with nanoparticles and nanomaterials is go after multiple sites of attachment on COVID-19, not just these spike proteins or S proteins, but also try to attach your molecule to the envelope protein E that you can see in this diagram, or the membrane protein M, where you can develop these unique nanostructured features to go in and attach to those regions. Go after multiple sites so that if a mutation occurs, and it will occur, it's already occurring, that COVID-19 is mutating, your nanoparticle strategy is still valid. So we know COVID-19 has multiple nanostructured functionalities. We also know that COVID-19 cannot withstand heat. Just like many viruses, there is some temperature at which, if you increase your temperature, you can disrupt the structure of that virus to keep it from attaching to a cell and then going in and replicating. So heat could potentially be a strategy. And I think we all know many nanoparticles, due to their exciting photothermal properties, you can externally increase their heat. You can generate through infrared gold nanoparticle photothermal properties that can heat up. So get that gold nanoparticle to attach to the virus, use infrared to heat it up, and maybe you can disrupt the structure of that virus. The other thing I listed on this slide is that COVID-19 has a lipid region. A lot of viruses have these lipid regions that can be disrupted, and they can be disrupted by generating reactive oxygen species. There are many great nanoparticles because of their small surface because of their large surface area to volume ratios, can generate a lot of reactive oxygen species. So if you can use a nanoparticle like that, generate a lot of reactive oxygen species after attaching to the virus, you can potentially destroy that membrane and again, destroy that virus so it cannot replicate. So I think there's a lot of things to look at in COVID-19. It's nanostructured, multiple nano functionalities, cannot withstand heat, and is susceptible to reactive oxygen species that really beg our attention to generating nanomaterials to fight viruses. So let me expand on this idea a little bit more. And a lot of these nanoparticles that I'm going to talk about have been used to fight cancer. They've been used to fight bacteria infections. They've been used for many applications. We just need to move those same nanoparticles into fighting viruses. And by the way, people have been looking at a long time at using nanoparticles to fight viruses. Zika, Ebola. There's a lot of research in this area showing a lot of promise when using nanoparticles to both prevent, diagnose, and also serve as a therapy. And I remember back at the start of my career, loved these high surface area nanoparticles, right? You decrease the size, and from some scanning electron micrographs are pictured here. You decrease the size of a particle to get so much more surface area. And that was a great thing. Still is a great thing because, again, you can have increased photothermal activity when you increase surface area. You can have increased magnetic properties. You can find ways to attach that cell, that nanoparticle to the virus. So that ability to create smooth kind of nanospherical structures was very important back then, and it still is today. But now, as you might know, we've been able to not just create smooth nanoparticles, but we can create particles like those shown on this slide. And actually, I brought some, I have nanoparticles with me at home. We are quarantined for the most part here in the United States, although we're returning, some of that leaving the house is returning. But I have nanoparticles with me at my house. So I wanted to show you some of them. This is one called ceria. So it looks like milk, but it's ceria or cerium oxide. And this is a wonderful chemistry to study to fight viruses. The reason is because you can make them look like these on this slide, with all of these fascinating junctions and extensions. And it reminds me of COVID-19, where COVID-19, you have S proteins. And then again, as I mentioned before, you have not just S proteins here in COVID-19, but you have these other regions that you can target, these membrane proteins or the envelope proteins. Well, if you're developing these ceria nanoparticles to have these kinds of multifaceted structures, they can get in there and they can attach to a membrane protein, or they can attach to an envelope protein and attach to that S protein. So again, you can use this idea of multifaceted nanoparticles to attach to many different regions on a virus, not just the S protein. So this ability of going from where we used to think about smooth nanospheres, but now you can make very rough nanoparticles, can help you treat viruses. We also know, as I mentioned before, that nanoparticles are very easy to functionalize. So whatever, if you're talking about, these ceria nanoparticles or whatever chemistry, we have well-established mechanisms to attach chemistries to these nanoparticles that can allow them to attach to a virus. We just have to know what should we go after. And I'll talk about that in a little bit. What functional group should you put on a nanoparticle to attach it to COVID-19? Fortunately, our virology friends have figured some of that out, that those of us in the nano area can use. We also know that we can make nanoparticles out of common everyday dietary elements. And here's another nanoparticle that we love, and you can see its nice pink orange kind of color, selenium. Selenium is a chemical that I think has huge potential in nanoparticle form. It's part of your diet. And again, you can functionalize it to attach to a virus. So obviously, why is that so important? Once the selenium nanoparticle attaches to a virus, disrupts its ability to attach to a cell membrane, it will naturally degrade and boost your immune system at the same time, because selenium is known to improve your immune system. So not only destroying the virus, but helping your immune system get back to normal is something you can think of when thinking of nanoparticles made out of dietary elements. As I mentioned before, you can take things like these gold nanoparticles. Here's some gold nanoparticles that we make in our lab. You can inject these, have them attach to COVID-19 or aerosolize them and breathe in nanoparticles so that they attach to your virus in your lungs. And then you can use an external near infrared to heat them up and again disrupt the structure of that virus through heat. And there's so many more attractive properties of nanoparticles. Here I showed you some ceria, some gold, some selenium. In the bottom corner, of course, something we'll talk about are these iron oxide nanoparticles, which are magnetic. So they can be used both for diagnosis to increase the concentration of a virus. If you can get these particles to attach to the virus and then use a magnet to separate them out of a solution, you can concentrate them, make COVID-19 easier to detect, but also in your body. You can use the magnetic power of nanoparticles to help disrupt that virus and keep it from replicating. So I really divided this presentation into a couple of short descriptions of how nanoparticles are being used for prevention to keep you from getting COVID-19, how it's being used to diagnose COVID-19, and then lastly, how can you develop a therapy around nanoparticles or nanomaterials for COVID-19. We are all, as an engineer, I'm certainly aware of PPE, right? As a chemical engineer, I definitely am aware of the importance of protecting yourself and wearing protective equipment. The field of PPE or masks, gloves, other protective type instruments, that field has, seen an explosion of using nanoparticles. There are companies now selling for many different areas in the world. These companies are selling masks, gloves that contain nanoparticles. Some of the nanoparticles that are in these masks, for example, are zinc oxide, silver oxide, copper oxide has also been used. And they've been used for two main reasons. One, as I think when you wear a mask, you are protecting other people. So if you're infected, but you're not showing symptoms and you have a mask, then that mask should you want it to keep the virus from going out and, infecting somebody else, having somebody else breathe that air. And that's, part of the reason why the World Health Organization has recommended people stay, 6 feet or longer away from one another because of this transmission that could occur through coughing or water drops that come out, saliva that come out when you cough. So if you have a mask that has these nanoparticles, the nanoparticles, first of all, decrease the pore size of your mask. And as we just talked about, these viruses are nano-structured. So you really need pretty small pore sizes in your mask in order to keep those viruses from going out. But the other reason, and perhaps, I guess equally as important, is these nanoparticles, zinc oxide, silver oxide, copper oxide, they can release reactive oxygen species. So if you have a moisture drop that comes out, gets trapped in the mask, these nanoparticles, high surface area, can release reactive oxygen species, destroy that lipid membrane in the virus. So even if a COVID-19 comes out, it dies essentially, or it gets disrupted so that it cannot replicate or get into somebody else and have a problem. So this ability for these nanoparticles to release reactive oxygen species to destroy the virus is another reason why they're being used in a lot of masks, and same with gloves. They're being added to gloves, silver, zinc, copper, to help destroy the virus should it get on your gloves and other things. I've even seen some clothing companies that are using nanoparticles and advertising them as a way to reduce COVID-19 activity on your clothes. So the field of nanoparticles in prevention has become quite popular, and people are showing some promising results. But another area that I want to talk about for prevention is everyday surfaces. And one of the things we have seen in my lab is you can actually take these, again, these are my selenium nanoparticles. We have a process that you can spray surfaces with selenium. They form nanoparticles on things like aluminum, which is what's pictured here, nano-selenium coated aluminum. And those selenium particles, if a virus gets on that surface, on that aluminum, that selenium will help destroy the structure of the virus. And this is because in some of these viral structures, selenium will actually compete with sulfur and some other chemicals and start changing the structure of that virus. So you could think about everyday surfaces being decorated, coated with selenium nanoparticles for disrupting that virus structure. Of course, the same thing can be said if you're coating a surface with zinc or silver oxide or copper oxide, where if a virus gets on that surface in a moisture drop, ROS or reactive oxygen species can be created to really help. The other thing that's going on in the field of surfaces is people, including ourselves, are nanotexturing those surfaces. And we're using nanotextures as a way to change the energy of that surface. And viruses, like everything else, have energy. So you can design a surface, if you understand what the energy of the virus is, COVID-19, you can design your surface to be super hydrophobic or reduce the absorption of that moisture drop, reduce the absorption or interaction of COVID-19. So there's a lot of examples of this, and highlighted on this slide are titanium, where you can anodize titanium. Titanium is used in many different everyday surfaces, create nano features. That in turn changes the wettability or contact angles on your surface, which in turn can keep something like COVID-19 from attaching. And I don't have a lot of time to get into the details of this, but you probably know nature has done this as well. And I used to talk a lot, still do, talk a lot about how nanotextures on insect wings can reduce bacteria attachment. Well, we've recently learned that those same insect wing nano features can reduce virus attachment as well. So many of these insects, many of these organisms have evolved to have features that can actually reduce microbial, bacteria, or virus attachment. So this whole nanotexturing idea to reduce interaction of viruses is something that is all over nature. So we're simply borrowing it and using this idea to reduce the spreading of something like COVID-19. And we have seen, through the years of our research here, you can see some PLGA polymers that were created to have micron features, some that were created to have nano features. And if you just look at the image D, this is an example of how a nanotextured surface can change the bioactivity of a protein. If this happens to be a study we did on fibronectin, where if it absorbs to a micron surface, if you look at that micron D image, these are AFM images, fibronectin absorbs like a blob. That's a non-bioactive form. Simply by changing that chemistry to have nano features, you can see the protein is more spread out. If you look at nano D image, the AFM image, same protein, but now it's spread. So you can not only change energy of your surface to determine whether that virus will absorb or not, but you can change proteins. And there will be proteins that come out if you cough in saliva. So you can change the bioactivity of those proteins, which in turn could create your surface to be more repelling to bacteria. And the last thing on this slide is we've actually developed an equation that can predict the height, the width, and the length of a nano feature to put on a surface to keep bacteria, to keep viruses from attaching, no matter if you're talking about a polyurethane coating that you put on wood, or if you're talking about a metal doorknob. We have this equation that can predict those kinds of features to keep something like COVID-19 from absorbing. So there's a lot going on in terms of prevention using nanoparticles. Similarly, there is a lot going on in our field for using nanoparticles to help with diagnosing whether you're infected with COVID-19. And I think we all understand our current problems with diagnosing COVID-19. It started out that you had to go into a hospital to get tested for COVID-19, and that led to spreading of COVID-19 in hospitals and really created an overwhelmed healthcare system, especially here in the United States. But the other, so certainly thinking of a diagnostic tool that you can do at home keeps you from going out and spreading it if you're positive. The other thing that's been a huge problem with testing is the number of false positives. In fact, I read a study from our Centers for Disease Control in the United States the other day that false positives can be upwards of 30%. So whenever we see the number of cases of COVID-19 and how it's going up, we have to take that with some caution, because we're not entirely sure our current diagnostic kits are accurate enough to say that number, to say that that's a true positive number of cases. So we have to do a much better job in terms of our diagnostic kits, making them at home so that you can use without going out, increasing accuracy of what we're measuring, increasing the speed. When also our conventional way of thinking of diagnosing viruses is to think of a PCR, polymerase chain reaction kit. These are not quick kits to determine if you have COVID-19. So, and that we have now kits and diagnostic kits that are quicker, but clearly we need quick, convenient, cheap, fast diagnostic kits in order to improve how we're detecting viruses. And again, we should have known that the next virus would have caused these problems. So we need to be much better prepared in terms of doing better diagnostic kits. So nanoparticles, however, are being used in all of these, and I'll show you a couple of quick examples where nanoparticles are being used in PCR, serological tests in which you're testing blood or solutions, looking at the presence of antibodies. Nanoparticles are being used to increase sensitivity of those tests. And certainly this last one, point of care kits, things you can do at home with determining if you have COVID-19 to increase sensitivity and reduce your need to go outside. So just a couple of quick examples. And again, these are some of my favorite nanoparticles. Here's my gold nanoparticles. Both of these examples on this slide talk about using gold nanoparticles, functionalizing them with regions that can attach to a virus, whether it's Zika or Middle East coronavirus. And here in these kits, you're taking advantage of the thermal properties, or I should say the plasmon resonance effect of gold. All of these nice electrons that gold has, and if an attachment occurs, if you get gold nanoparticles to interact with quantum dots, the example on the left here, you can amplify your signal because of all the electrons that gold has interacting with a quantum dot that has detected and attached to a virus. You can now increase sensitivity. And the sensitivity levels that people are reporting are amazing, down to kind of the femto level. So you have this ability to really enhance signal intensity so that you can determine small numbers, small amounts of a virus. Here's examples of other diagnostic kits where gold nanoparticles are being used together with silver. And we all have known for a while how silver can help with Raman spectroscopy, enhance signal of something like Raman spectroscopy to look at weak signals, amplify them because of all of these electrons, that are around silver. So same idea. You can have a diagnostic point of care kit, drop a sample onto this kit that has gold and silver. And if an attachment to the virus occurs, your signal can get amplified because of these electrons that you have, significantly greater electrons you have because of the small particle size, increased surface area. And the last example here before moving into some exciting therapies are iron oxide. I showed these before, some iron oxide nanoparticles, where if you have a solution of your virus, or in this case, it's an example for bacteria, you can use a magnet as shown in B. Once those nanoparticles attach to the virus or bacteria, use a magnet to concentrate those microbes to then be able to determine and get a better signal because you've concentrated those microbes, the virus and the bacteria. So let me move on to what I think is the most exciting area, which is therapy. And this is not to discount, there is wonderful work, as we all know from cancer, the field of theranostics, where nanoparticles really have driven this field where you can develop the same particle to both detect and serve as a therapy. So there are active research areas in theranostics for viruses as well that are going on. But let me start just taking a step back and thinking of therapies, if you're infected with COVID-19, that deal with self-assembled nanomaterials. And we all know what self-assembled materials are. These are chemistries that you create synthetically, you add them to bodily fluids, and they organize in a very controlled way. So they assemble in a very controlled way. We happen to be looking at two type of self-assembled nanomaterials that can self-assemble around COVID-19. So it serves as a blanket or a jacket where you're coating the virus to in turn keep it from attaching to a cell and entering the whole replication process. So very quickly, two of the self-assembled materials that we like. Here's one of them. We call it, it's basically a peptide amphiphil, cationic peptide amphiphil. And you can see the chemistry in A, where we put in there some chemistries to help it self-assemble. And then we also put in there some chemistries that our collaborators, our virologists, have told us, if you develop this chemistry, you can attach your self-assembled molecule to the virus. And again, here we're going after multiple sites, not just the spike protein, but we're going after the region in the membrane protein, in the envelope protein of the virus, going after multiple attachment sites, and then have our molecule self-assemble around that virus. The other thing we have learned in these studies, and you can see in this drawing on this slide in B, is a fiber works better than a sphere. And I think this intuitively makes sense. So we are developing molecules that self-assemble into a fiber because we want it to wrap around the virus. A particle will just, stick to the virus, and surely it can influence its behavior, but not as much as a fiber that can start wrapping around a virus. So again, here's the structure of the virus. You can see in electron microscopy and some of the very specific sequences that we're looking at in chemistries to construct this molecule. We have a long history of looking at this molecule for killing bacteria. Here's MRSA, the antibiotic resistant version of Staphylococcus aureus. And we have seen that these peptide amphiphils will self-assemble and poke a bacteria, causing that membrane to rupture and causing the inside, obviously, of the bacteria components to come out and killing that bacteria. We've also seen that this works for multi-drug resistant E. coli. So we're pretty excited about using these for bacteria. And I'll show you some of the results in viruses in a second, but let me also explain our second self-assembled molecule that we're studying. This we call helical rosette nanotubes. We made these molecules in collaboration with Professor Hisham Faniri. And these molecules are essentially DNA-based pairs, guanine and cytosine, that we functionalize together. And you can see, again, the specific chemistry on this slide. When 6 of these guanine cytosine regions get together, they form a tube. In fact, they'll further self-assemble in the Z direction and start twisting to really give you this molecule that can enwrap or envelop a virus. And again, with these rosette nanotubes, just like the peptide amphiphils, you can functionalize them. So in this previous slide here, we're showing lysine as the functionalization strategy in green, but we can put on here a number of different peptides that will attract it to the virus, to different regions on the virus. So we have a history of looking at these molecules in tissue engineering approaches. Here's just a quick example for bone and one for cartilage, where we've functionalized the helical rosette nanotubes to increase bone growth. And we've also functionalized them to increase cartilage growth. We've done some animal studies to show that this can work. But I think the other important thing that we learned about these helical rosette nanotubes is we get significantly reduced inflammatory reactions surrounding these molecules. We don't see the inflammation that you typically see when using hydrogels, for example, for healing cartilage, or using titanium to grow bone. And that led us to thinking a couple of months ago, there's a big problem with COVID-19 and your immune system response. We often call it, and the field calls it, the cytokine storm. This is where your inflammation, your inflammatory system is producing cytokines, growth factors, proteins that overdo it. They over stimulate your immune system. So we not only need materials that can attach to a virus to deactivate them, but we also have to deal with your immune system response. So if you have a nanoparticle or a nanomaterial, like we think we might here, that can both attach to a virus as well as deactivate your immune system, you can accomplish both the cytokine storm and keeping that virus from replicating. So real quick, here's just a spotlight of some of our results for the peptide amphiphils, the helical rosette nanotubes. We functionalize them again to attach to COVID-19. I know a lot of people ask, what groups do you know to use? And let me point you to this reference on this slide. There was an excellent paper that came out of India back when all of this started in China. And this paper looked at the mutations in COVID-19 compared to other viruses. And it lists very specific peptides, very specific functional groups that are different in COVID-19 compared to these other viruses. What we did is we developed complementary chemistries to attach to our self-assembled molecules, which in turn can then attach to the virus. So these kinds of papers were so important for us to know the biology, the chemistry of the virus, so that we can identify complementary chemistries to attach to the virus. The second bullet on this slide is also important. Without doing one experiment, we used models. So this website that is listed up here was created by a group of computational modelers. And this computational model tells you the strength of which your functional group can attach to COVID-19. So clearly you want as strong of an attachment as possible. So before we did any experiments, we basically ran through this model, used this website to tell us if we talk about the peptide amphiphils, the APNPs, or we talk about the helical rosette nanotubes, which functional group gives us the strongest attachment. And we learned a number of things by going through these models that I don't have too much time to talk about, but the one is we learned, using a nanomaterial improves attachment strength compared to just using the peptide or the molecule alone. So the self-assembled molecule helps that peptide attach to the virus. That's great news, right? That attachment strength goes up. It helps in a competitive environment, that self-assembled material attach and that peptide attach to the virus to deactivate it. The other thing we learned is if you have multiple functionalizations, again, not just going after the S protein, going after these other regions in COVID-19, your attachment strength goes up. And that makes sense because you're going after multiple sites. So your molecule is attractive in a greater sense to the virus and will attach to it in a greater manner. And that helps deactivate it. Of course, we've also been looking at nanoparticles, and I'm almost out of time here. So let me, just say quickly that many people, including ourselves, have been looking at, chemistries like PLGA, that in nanoparticle form, you can deliver a drug. Remdesivir seems to be the best current FDA-approved drug to treat the symptoms of COVID-19. So clearly, we can take the wealth of knowledge we know about PLGA nanoparticles lasting a long time in your circulation, prolonging drug release, targeting the release of this drug. We can load PLGA with remdesivir or some of these drugs to really help the symptoms of COVID-19. This has been done for HIV, another virus that obviously has in the past created a lot of problems. In terms of vaccines, you can see a couple of studies that I've highlighted here for influenza, where you're using lipid nanoparticles, really helping build up the antibodies that are necessary for a vaccine. I would also mention that one of the earliest nanoparticles used in vaccines was alumina. Al2O3 nanoparticles are used as a vaccine, and that small particle, which has great surface area, can invoke a greater immune system response to develop a vaccine. So the same idea here, where you're using small particles to get a large immune system reaction, large number of antibodies to be produced, holds true for vaccines as well. And of course, back to my friend selenium, I have to show it to you again because I love this material so much. Making these nanoparticles for a therapy out of healthy components to your diet can both help your immune system and treat the virus at the same time. So here's a study in which selenium nanoparticles, this was not conducted by our group, selenium nanoparticles were functionalized with an antiviral agent to treat influenza, and selenium was chosen because it is a natural part of your diet and showed a lot of efficacy. And of course, the other example here you can see are iron oxide nanoparticles, which helped in terms of therapy for influenza as well. So I'm going to skip by this part in the interest of time, but certainly there's a lot of value of looking at iron oxide nanoparticles to fight viruses, which I think I've alluded to throughout this presentation. And let me leave you with one thought. And that is, we need to, if we think about nanoparticles fighting COVID-19 or using them in medicine in general, it doesn't make sense for us to use toxic chemicals when making these nanoparticles. We're using them in medicine, we're using them to help people's health, but yet many processes that make these gold nanoparticles involve the use of toxic chemicals. So we as a field need to think about non-toxic chemicals, non-toxic approaches to making healthy nanoparticles to be used for killing viruses. And we have been doing this in my lab, where we don't use synthetic conventional chemistry to make these particles. We're actually using cells to make these nanoparticles. So we add metal salts, we add components to cultures of bacteria to make nanoparticles. And the interesting thing, not only is it not toxic because cells are making them, not your toxic chemicals, but we're also seeing that these particles are more effective at killing viruses, at killing bacteria. And to make a long story short, if you look at image C and D, when the bacteria make these nanoparticles, they make them with a protein coating. That protein coating makes them healthier for your body. And that protein coating also helps target that bacteria, that nanoparticle, to the bacteria that made it. So quick example, here is selenium again, nanoparticles that were made by a bacteria, E. coli. These particles are more effective at killing E. coli than healthy bacteria in your microbiome. And that's because this protein layer surrounds the nanoparticle and targets it towards that bacteria. So a win-win, healthy approach, not using toxic materials, but also a way to make the particles more effective. So some of the raw data I don't have time to go into, but let me, quickly conclude that I'm seeing a lot of activity using nanoparticles for preventing COVID-19, for helping to diagnose COVID-19, and certainly for therapies, as well as a combination of all three. I think their small area, surface area, amplifies their properties to target those viruses, multiple sites of targeting, to be able to be controlled externally, to disrupt the function of that structure of that virus, and ultimately to be formulated out of healthy components and not, toxic components to your diet. So I thank everyone for paying attention for this presentation. I hope that everybody's staying safe and being healthy. I certainly want to thank some of the funding agencies of the work I talked about, and definitely my research lab. I know we have only limited time for questions, so please reach out to me on the web. My email is right on this slide. We love collaborating. We love thinking about how these nanomaterials can disrupt viruses in this new area for us to really help us all return back to an active lifestyle in terms of research, in terms of social gatherings, et cetera. And my last comment, COVID-19 is a severe pandemic, but there will be another one, and we have to be better prepared. We have to create platform technologies and not automatically think of waiting a year and a half for a vaccine for that next virus. So thank you very much. Geetha, I don't know if we have time for questions or if I talked too long as usual.
First, I would like to thank you. It's always a pleasure to listen to your talk. It's very interesting to note your long-term experience in the field of nanomedicine. You are able to give an overview of how nanomaterials can help in diagnosis, prevention, and therapy. The good news is there are around 600 participants viewing your listening to your lecture, and we are bombarded with around 98 questions. If we wait 2 more minutes, maybe it will cross 100. And so what we plan is we will collect all the questions and send to you. Apart, they can also contact you as you have given your email address. The most of the questions which were being like every 1 minute, one more question was coming, and I was going through, and most of the questions as you were talking, you were answering it. So partially the questions are like that. And the most important I feel is everybody was concerned about the toxicity of the nanoparticles you are using. And they also were saying like selenium, we have some reports on the toxicity of the selenium. So what is the dose level, the toxicity level, what is the retention in human, how it is excreted. So most questions were towards that direction. And the second question, some questions like whether we can use biopolymeric nanofabrics as a good candidate for our masks. So can you answer? Then I will get back to the next question if you have time.
Yeah, sure, absolutely. And toxicity is a huge area of concern for nanoparticles for any application, cancer, viruses, whatever you're using those nanoparticles for. And that's why I'm so happy to see the field moving towards more healthy nanoparticles. Gold is wonderful, and it's really served as a foundation for the nanoparticle, nanomedicine area. but things like selenium, things like iron. Iron is part of your natural diet too. And many COVID-19 patients are anemic. They're actually low in iron. As you know, many people who are suffering greatly from COVID-19 already have underlying health problems. So if we can start thinking about using chemistries that are much more healthy to your diet, that can increase iron levels for an anemic patient, that can boost your immune system, that to me is the way to go, not just for viruses, but for everything. We are seeing in our studies that you need a microgram or less quantity of selenium, for example. I'm remembering some of our results to deactivate COVID-19. Now, of course, this depends on how much of the virus you have, but it's a very low concentration you need. And I think the reason it's so low is, first of all, we're functionalizing selenium to attach to the virus in multiple regions. We use that computer program to find the highest attachment strength possible, but also selenium nanoparticles and most nanoparticles can be aerosolized. And the virus is right here. So if you're breathing in these nanoparticles as a therapy, you don't need as many because you don't have to go through the entire systemic circulation. Now, there is some evidence emerging that the virus gets into your bloodstream and that is causing for some cardiovascular problems. So in those situations, you will have to think about a higher concentration because now it's systemic. But if it's in the lungs and we can catch it early enough, you don't need that high of concentration. And you know, delivering 1 milligram in total of selenium nanoparticles is well below, orders of magnitude, you know, below the dietary intake allowance that you need for selenium every day. So definitely, you know, we need to pay attention to more healthy chemistries when using nanoparticles across the board. I love the idea of masks made out of, I think it was also healthy materials, right? You know, and the key thing with the masks is that pore size. And if the virus gets on there, you know, how frequently should you wash the mask or bleach the mask, right, to kill the virus? So if you can develop a mask out of these natural materials that can both capture the virus and then deactivate it, your concern, you know, with reusing it goes way down. And I think that's so important and something that the field of nanotechnology, nanofibers can really play.
Okay, as a researcher, I mean, you're the master in the nanomedicine. Like everybody now wants to work in the field of COVID. This is my question. And always prevention is better than therapy. So I think like a lot of people have to go into the direction of prevention by finding a spraying or coatings and all that stuff. So what is your view on this? And many don't have an access to the study of virus effect virology department. So what will be a good start for young people who are listening to your talk to begin with and work towards for the pandemic situation?
Yeah, absolutely. You know, I think prevention is so important, and you know, prevention is working, right? We often say our best strategy today is not medicine. It's separating yourself from somebody else, social distancing. That is our most effective strategy right now. So certainly that's important. However, I also realize, you know, not everybody will wear a mask. Not everybody will social distance. You might know, you know, we have some rioting going on in the United States right now. People are not social distancing in those riots. So I also realize you have to have a very comprehensive strategy, not just on prevention. And I think, you know, the best thing we can do for diagnosis and therapy is to combine them. Same material that can do both, as well as make it as easy for people to use as possible. And that, you know, I was quite surprised when all of this started in the US in March. It really started getting out of control. The message was, if you feel like you have symptoms, you still have to go into a hospital and get a test. It wasn't until late April that we actually had kits that you could buy and be delivered to your home. You get a sample, usually your saliva, you put in this kit and send it back. I mean, that was a good month that we lost in terms of restricting people from going out and spreading it even further. And in those kits, the more we can use nanoparticles, the more we can increase sensitivity of a measurement, and hopefully the more accurate our measurement can be. So I think my advice, you know, for people who want to get into this field, a lot like what we did, right? We had a small history, and I don't mean that because of nano, but we had a small history of looking at viruses in the past, influenza, norovirus, things like that. But when COVID-19 came out, our first question was, what is its structure? We need to know the chemistry, because if we know the chemistry, we can design nanoparticles to diagnose, to treat, you know, even to put in masks. If you have the nanoparticle that's functionalized for COVID-19, it can help capture that COVID-19 and keep it from spreading to somebody else and deactivate it. So we needed to know the structure. That now exists, thankfully.
There is one question: which shape of nanoparticles is the most apt for the virus killing?
Yeah, so the one that I showed, that the Syria, right? And this, you know, is not just because of the chemistry. You might know Syria has a great ability to release reactive oxygen species. So the ones with those facets really get into the virus. We've seen that they have great attachment strength, great attachment to COVID-19. So, and the other part, for people just entering this field, is to use these models. These computational models, the website that I showed, really give you a way to do something without being in the lab working with COVID-19. You know, are your functionalized regions helping attach to the virus? So get to know the structure, look that up, you know, figure out how you can design your nanoparticles appropriately, and then model it. And then you'll have a pretty good idea to then collaborate potentially with somebody who can culture COVID-19 and see if your approach works.
Yeah, that's a great idea. Due to want of time, I would like to conclude this session. Thank you once again, Tom, for coming forward to present this. See you.
Can't wait. Yeah, can't wait to see everybody in person.
Over the session. Thank you. Thank you so much.
Thank you.
Over to Loganathan.
Now I would like to invite Dr. Sudhakar Pichai, Swansea University, UK.
Hello. Okay, it's a time to formally thank to all. Good evening to all. First of all, I would like to thank Professor Thomas joining from States during this challenging time. I enjoyed your talk. You covered the panoramic view of a complete nanoparticle utilization in how to tackle the COVID. Thanks a lot, Professor Thomas. Okay, my sincere thanks to VIT management and Swansea University, particularly College of Engineering and Swansea University Medical School, for extending a warm support to conduct this event very smoothly. My hearty thanks to Professor Geeta Manivasagam, organizing chair of this event, and of course, Dr. Loganathan, Indian convener of this event, and beloved organizing committee members from Center for Biomaterials, Cellular and Molecular Theranostics, so-called as CBCMT, from VIT for their tireless commitment for the last few weeks. Without your support, we couldn't make this Indo-UK virtual conference successful. Okay, now on behalf of the organizing committee, we would thank to all the speakers for your valuable contribution, and we appreciate chairpersons of each session for their time and commitment. Our sincere thanks to the esteemed panelist members, Dr. Ravichandran, Director, NIPER, Kolkata, Professor Krishnan, Honorable Vice Chancellor of Madurai Kamarajar University, Dr. Taslima Arif, C Camp, Bengaluru, Dr. Kanesh Pandian Namachivayam, from Kyoto University, Professor Vengat from Swansea University Medical School, and Dr. Swaminathan Sethu, Narayana Nethralaya Foundation, Bengaluru, for sharing their views and comments on challenges and management of COVID-19. I think this panel discussion accelerated the academia, industry, policymakers, stakeholders network for tackling coronavirus pandemic disease effectively. We appreciate all the delegates submitted the abstract, and hearty congrats to all the presenters in the oral and poster presentation session. It was exciting to see around 600 to 900 participants constantly joined all the invited talk session and raised very thoughtful questions. As you know, during this coronavirus pandemic lockdown period, we are facing a lot of challenges in our routine. Importantly, sustainable internet connection is the great challenge during this time. But our participants came across these technical hurdles and supported us gratefully. In fact, many of the participants were joined us from interior places of India. Also, we would like to thank the overseas participants for more than 25 countries. Though they have a time constraint due to the different time zone, they showed amazing support for us. We hope you enjoyed all the sessions. If you have any inconvenience during the event, please accept our apologies. Once again, on behalf of the organizing committee, both from VIT and Swansea University, thank you so much. Take care and stay safe.
Thank you all. A couple of announcements for the participants. Certificates for the participation, oral presentation, and poster presentation will be issued by next week by email. Since we have 3,000 participants, please give us a time for 1 week time. We will respond to you in an email. Thank you all. We want to then tomorrow we are planning to display the poster in a YouTube live video. The link will be shared in our conference website as well as in the conference Facebook page. Please stay tuned and please stay safe, stay healthy, take care. We shall be closing now formally. Thank you all.